Okay, so this is going to be a hodgepodge of different papers that I have looked over, and I don't really have a good, I guess, what would the word be, outline for what I'm going to talk about, but my general thesis is that I believe that iron dysregulation is playing an extremely large role in these pro-coagulatory effects that we are seeing in COVID-19 patients today. And, excuse me, one of the reasons why I think that is because interleukin 6 has been greatly upregulated in COVID-19 patients, and there are many different things that can cause the upregulation of interleukin 6. However, I want to draw our attention to, I really would like to draw our attention to iron. So the first thing that I want to mention is that iron can, in of itself, induce interleukin 6 by the production of free radicals and by the production of pro-oxidant species within the body. So in your body, you have proteins that sequester the iron ions. These are such as ferritin, which is the storage protein of iron in your body. You also have transferrin, which is the protein that will take two ferric ions and then sequester them so that it can be transported throughout the body without reacting with other proteins. Because iron ions are very reactive. They are redox reactive, so they can catalyze redox reactions or reductive oxidation reactions. They can catalyze the production of free radicals. They can catalyze the production of superoxides. And so all of these things can in of themselves create a pro-inflammatory environment, which would cause interleukin 6 production. However, what I want to stress is that the upregulation of interleukin 6 is not, I believe, sufficient to describe what we are seeing right now. So here I want to make mention that the induction of interleukin 6 by coal-containing bioavailable iron is induced through both hydroxyl radicals and ferril species. So what is a hydroxyl radical? So radicals are, in terms of chemistry, radicals are molecules that do not fulfill the octet rule. So they will have an unbound electron of some form. So for instance, you could have the hydroxyl radical, which will have an unbound electron on the, or should it say an unpaired electron on the oxygen, and it is a neutral form of the hydroxide ion. So because this electron is not paired, and because the octet rule has not been satisfied, this radical is very reactive. It can destroy a lot of the proteins and DNA within your body. So your body has developed defense mechanisms to sequester or remove these hydroxyl radicals. And one of the side effects of these radicals, or one of the ways that your body communicates that there's oxidative stress, is to go through this pro-inflammatory cytokine pathway, which in of itself is beneficial and there to protect you. So this is to protect you from the hydroxyl radicals. However, if it gets dysregulated, you can have some problems arise. So, sorry, there was a tornado warning earlier today, which is why I'm a little wet, and it's currently raining a lot, so I actually had to seek shelter earlier tonight. Regardless, this is what a hydroxyl radical is, and here we have a ferril species. So what is a ferril species? This is a high valent iron, or it is iron that is above the 3+ state. So iron, we normally think of it as being in the 2+ or the 3+ oxidation state, but iron can in fact become even more oxidized. So here this will be iron in the, what is this, 1, 3, 6, yeah, this is in the 4+ state. So this is what is called a high valent iron, and this in of itself is also very reactive as well. So when there are people who work in coal mines and they breathe in, say, coal dust that has a lot of iron, especially bioavailable iron, this can result in a very pro-oxidative environment. So here I'll just read through the abstract. So coal mining causes health problems such as pneumoconiosis, and we have previously shown that prevalence of pneumoconiosis in workers from various coal mine regions positively correlates with levels of the bioavailable iron, also known as BAI. So looking at a cohort of different miners, you can look at the iron, the bioavailable iron that is in those coal mines, and you can determine what is the likelihood or the severity of this pneumoconiosis. I can't pronounce it, I apologize, but pneumoconiosis. So in the coals from that region. So in the present study, the nature of reactive oxygen species formed by bioavailable iron in the coals and its mechanisms of the induction of biological responses were investigated. So the human lung epithelial cell line, also known as, this one is A549 cells, these were used to examine the induction of interleukin 6, which is a pro-inflammatory cytokine. So let us look up what interleukin 6 is. I wonder if there is an image that you can see. So yeah, if this loads, the internet isn't necessarily the best at this time. So this is an image of interleukin 6. So it's an it can act as both a pro-inflammatory cytokine and an anti-inflammatory myokine. So this, if it comes from the muscles, it can be anti-inflammatory, but for in most situations, we call it pro-inflammatory because it's, at least in the epithelial cells and in the normal tissues of the body, it tends to be pro-inflammatory. So let us go. So this is a pro-inflammatory cytokine usually, and which is known to play a crucial role in the development of pneumoconiosis. And we found that levels of interleukin-6 protein, as well as its mRNA, were significantly increased in the cells treated for 24 hours with 20 micrograms per square centimeter of the BAI, bioavailable iron-containing Pennsylvania coal. For example, we observed a 6.7-fold increase in interleukin-6 production. So this is the interleukin-6 that's being produced by these epithelial cells that are just unfortunately being triggered by this bioavailable iron. And this bioavailable iron, which if it goes through your digestive tract and it's being sequestered by proteins so that it's not reactive anymore, that is healthy and that is good and necessary for you to have proper iron homeostasis in your body. But if you're just breathing it in, you do not have those same mechanisms in place, and therefore you can start generating more of these reactive species, which thus will produce interleukin-6. So, and when it was treated with Utah coal, which contains low bioavailable iron, it was only a 1.9-fold increase versus the 6.7-fold increase. So the enhancing effects of interleukin-6 by the Pennsylvania coal was similar to that caused by hydrogen peroxide. So superoxide dismutase, which is an enzyme that will remove superoxide from the system and turn it into hydrogen peroxide. So that's superoxide dismutase. So hydrogen peroxide is actually less damaging than superoxides are. There's also catalase, which catalase turns, what is it? Yeah, it turns hydrogen peroxide into water and oxygen. So by having both superoxide dismutase and catalase, you're able to turn superoxides into oxygen and water. There's also N-acetyl-L-cysteine, or NAC, which is a antioxidant that is, I believe, a cofactor for one of these enzymes. These all had an inhibitory effect on the Pennsylvania coal-induced interleukin-6 formation. So these antioxidant effects are truly lowering the interleukin-6 levels. However, catalase had the least protective effect as compared to the superoxide dismutase and the N-acetylcysteine. So our results indicate that BAI, bioavailable iron in the Pennsylvania coal, may induce interleukin-6 through both feral species, which is through iron autooxidation, and hydroxyl radicals, which is through the Fenton-Haber-Weiss reaction. So this is basically when oxygen reacts with iron, it ends up catalyzing the production of superoxide, and when hydrogen peroxide interacts with iron, it catalyzes the production of hydroxy radicals. So yeah, this is all extremely important to how to phrase it. These antioxidant enzymes and chemicals are necessary for removing these pro-oxidants or these oxidative radicals. And let me look what N-acetyl-L-cysteine does explicitly, because acetylcysteine, also known as NAC, is a medication that's used to treat this. And let's see if they have a, you know what, they don't have a chemical reaction right away, so I'm not going to look it up. Regardless, just keep in mind, this is one of the proposed mechanisms. So there is that. What I also want to talk about is the iron-induced interleukin-6 gene expression. There's possible mediation through the extracellular signal-regulated kinase and P38 mitogen, MAPK, or mitogen-activated protein kinase pathways. I'm not going to go too far into detail. Just know that this is another paper describing the fact that increased iron can cause the production of interleukin-6. So just keep that in mind. Okay. So with that, I will divert my attention to the Cell paper that I did a video on earlier today, where we were talking about Galectin-3 binding protein being upregulated in COVID-19 patients. So what does this Galectin-3 binding protein have to do with anything? Well, one, Galectin-3 binding protein is also associated with the production of interleukin-6, and this process is mediated by it and its interaction with Galectin-3. So remember, this is Galectin-3 binding protein. So Galectin-3 binding protein and Galectin-3, they are going to co-localize in the areas of the endothelial cells, of the vasculature of the, how to phrase that, in the veins and arteries, predominantly the veins, I believe. And this then convinces or puts you into a prothrombotic state. And so this interaction is actually what convinces some of the neutrophils, which are granulocytes. So these granulocytes are white blood cells that have a bunch of little granules inside of them with different chemicals in them, such as that are cytokines, right? So the interaction between Galectin-3 binding protein and Galectin-3, this convinces those white blood cells to bury themselves into the endothelial cells. And once those cells are buried into there, it convinces or causes the upregulation of interleukin-6. So that is how Galectin-3 binding protein is associated with the production of interleukin-6. However, the paper that I described earlier today, that you can go watch that video as well if you want, it said, or it showed, that in mice where you are inducing thrombosis through whatever model mouse they had, there was no increase in Galectin-3 binding protein. There was only an increase in Galectin-3, but there wasn't an increase in Galectin-3 binding protein. That was in that particular model. However, what we have here, what we could also presume and basically figure out, that if you have an excess production of Galectin-3 binding protein that is in the serum, because remember, this is in the serum, if you have an excess, then that might as well also create a prothrombotic response, or at least it might heighten a prothrombotic response, so you would be more likely to clot. So what I want to do now is I'm going to talk about how this Galectin-3 binding protein was upregulated, but I also want you to keep an eye on this albumin. This albumin is downregulated, and so what low levels are associated with acute phase responses and observed in critical COVID-19 patients. So what is albumin again? This is a general plasma protein. So this is a globular protein, so it forms a glob basically, and it's the most common of which are the serum albumins, and all the proteins of the albumin family are water soluble, moderately soluble in concentrated salt solutions, and experience heat denaturation. So albumins are commonly found in blood plasma and differ from other blood proteins in that they are not glycosylated. So this is a non-glycosylated There are no sugars on this. So substances containing albumins are called albuminoids. So a number of blood transport proteins are evolutionarily related to the albumin family. So just know that that is what this does. And so albumins, in a less strict sense, can mean other proteins that coagulate under certain conditions, but that's a different strict definition. So let's see, is there, yeah, just know that this is a protein that's floating around in the body, but if you have severe COVID-19, you actually have less of it in your plasma. So just keep in mind that is also what's happening, and I will reference this later. So with that, I want to talk about a little bit of these unusual clots. So people have been talking about these weird clots that are forming in these people with COVID-19. Well, here I want to discuss how we have known for a significant period of time, since 2013, we have known that iron in of itself can cause unusual clots. It can cause unusual clotting, so not normal clotting. So here, unusual clotting dynamics of plasma supplemented with iron 3. So I'm just going to read the abstract of this one. So iron salts are used in the treatment of iron deficiency anemia. So diabetic patients are frequently anemic, and treatment includes administration of iron. So we give iron to diabetic patients frequently because they suffer from anemia. Anemia patients on hemodialysis are at an increase of thromboembolic coronary events. So just blood clots that cause embolisms of the heart. And this is associated with the formation of, dense fibrin clots. So they aren't calling this a normal clot. This is explicitly a dense fibrin clot. And this is resistant to fibrinolysis, which is the enzymatic breakdown of fibrin in blood clots. So basically, in your body, you have a protein called fibrinogen. Fibrinogen is synthesized in the liver and some other cells, and then it's floating around, and I believe it's mostly in a lot of the thrombocytes or a lot of the platelets, and I think it's also just floating around in solution as well. But fibrinogen. So you have this fibrinogen that's floating around. So it's a glycoprotein complex, and it's made in the liver, and it circulates in the blood of all vertebrates. So yeah, I was right, it is circulating in the blood. And during tissue and vascular injury, it is converted enzymatically by thrombin into fibrin, and then the fibrin-based blood clot. So basically, this fibrinogen gets proteolytically cleaved by this enzyme called thrombin. And after it's been cleaved by thrombin, then it turns into when this fibrinogen gets cleaved by thrombin, it gets turned into fibrin. And then fibrin is very, very easily will polymerize with other fibrin molecules, and this is what forms a clot. So this clot is going to have a bunch of these fibrin molecules that are polymerizing with each other to form this much larger macromolecule, if you will. So just keep that in mind. That is the general principle behind clot formation. So we can sometimes, if these diabetic patients are given iron to help with their anemia, every once in a while they will be, or I should say they are at a higher risk of creating these dense fibrin clots, and these dense fibrin clots do not easily degrade by fibrinolysis. So moreover, in chronic kidney disease patients, the high Labile plasma iron, Labile meaning that it's unbound, if I'm remembering correctly. Let's look this up. So what is Labile plasma iron represents a component of non-transferrin bound iron. So yeah, I was right. So this is iron that's in the blood that is not bound by transferrin, which, remember, transferrin, as I was talking about earlier, this was the protein that is involved in transporting iron throughout the body, and it will transport these iron 3 positive ions throughout the body and protect your body from the iron, because iron is so reactive. So if you have iron that is not bound to transferrin, that is considered labile plasma iron. So this labile plasma iron levels associated with iron supplementation are involved in complications found in dialyzed patients, such as myocardial infarction. So what is that? This would be a heart attack. So the aim of the present study was to investigate whether iron treatment is involved with the formation of fibrin clots, and clotting of citrated plasma supplemented with iron 3 positive was investigated in thromboelastometry and electron microscopy. So don't worry too much about that. So the results revealed that iron modifies coagulation in a complex manner. So iron chloride or ferric chloride stock solutions underwent gradual chemical modifications during storage and altered coagulation profile over 29 days. So this iron, after it's been added, the iron itself is reactive. So it's going to turn into a bunch of other different types of iron species while it is in solution. So this is suggesting that iron 3 positive interacts with both the proteins of the coagulation cascade as well as the hydrolytic iron 3 positive species. So iron extends clotting of plasma by interacting with proteins of the coagulation cascade. So the proteins of the coagulation cascade, whenever you clot, you actually have a bunch of different proteins that are all regulating each other to make sure that you are only clotting when you're supposed to. However, if this iron is screwing up a lot of those coagulation cascade proteins, this process could be theoretically become dysregulated. So there is that, and then iron 3 plus and/or its hydrolytic species interact with fibrinogen and/or fibrin. So it's interacting with either fibrinogen and fibrin, which is changing their morphology and properties. So remember how I said fibrinogen gets catalytically converted into fibrin through thrombin. Well, there is the possibility that these iron is actually directly interacting with fibrinogen and possibly causing it to polymerize, or it might be hydrolytically broken up into fibrin without thrombin being involved at all. So we need to be aware that that might be happening. So, and this is changing their morphology and properties. So in general, iron or ferric chloride weakens the fibrin clot while at the same time precipitating plasma proteins immediately after application. Precipitating plasma proteins. What is a plasma protein? Oh, albumin. Albumin in of itself might be getting precipitated by this iron 3 plus. So iron 3 plus or its derivatives induce the formation of insoluble coagulums in non-enzymatic reactions. Interesting. So including, what is this? Albumin and transferrin. Oh, you know what else I forgot to mention? What else is upregulated? Fine. Oh, sorry. Shift. Well, it was, oh. What is this? Transferrin is another one of the proteins that is upregulated. So our analysis associates several proteins with COVID-19 severity that have not been previously associated with the infection of the host response. These include CD14, this is the Galectin 3 binding protein that we were talking about earlier, and all the way down here, oh yeah, transferrin. That's being affected in some way as well. Now, what I would presume, we know that transferrin and ferritin, which is also elevated in COVID-19 patients, transferrin is actually going to be upregulated by the presence of iron. So is it possible that the presence of labile iron is causing more transferrin to be synthesized and produced? Possibly. So let's go back to this. And also this transferrin might, after it's being upregulated, it might be becoming precipitated in some way. So anyways, iron plays a role in coagulation and can precipitate plasma proteins. The formation of coagulums resistant to lysis in non-enzymatic reactions can increase the risk of thrombosis or excess clotting, and extending clotting of plasma can prolong bleeding. So that is one way that iron 3 plus can cause clotting. And then there was this paper that came out a little bit later called, this was December 2013, this was June 2013, so differences in the morphology of the fibrin clots induced with thrombin and ferric ions and its pathophysiological consequences. So here, oh, you know what? Oh, sorry, I lost. So here we have another paper that is describing the fact, and I actually lost access to this somehow. I used to have access to this, but apparently not anymore. So here is just another paper describing that there is some different morphology to the fibrin clots that are induced from thrombin, and there's a difference from that and the ferric ion-induced fibrin clots. So there is going to be, theoretically, a mechanism that does not even require thrombin to cause these fibrin clots or these dense fibrin clots from forming. So, and it also has pathophysiological consequences. So, yeah, I'll just read this, and then I'm going to go over to another. I'll probably go to, this hemostasis paper. So, the activation of blood coagulation leads to the formation of thrombin that in turn converts the soluble plasma fibrinogen into the insoluble fibrin clot. In healthy individuals, fibrin is effectively degraded. However, in prothrombotic states, proteolysis of fibrin clots are often delayed or even inhibited. So if you're in an extremely prothrombotic state, the destruction, the proteolysis or the lysis of fibrin is inhibited, and it's very difficult to get rid of them. And this is associated with altered fibrin structure. We have previously shown that in inflammatory conditions like stroke and diabetes, this fibrin forms dense matted deposits or DMDs. Although there are several factors that modify fibrin structure and delay fibrinolysis, in these conditions, no mechanism is yet known to be responsible for a persistent presence of thrombi or blood clots in the coronary and or cerebral circulations. So it seems, therefore, desirable to better understand this phenomenon in order to improve the effectiveness of thrombolytic therapies. So, and then thrombolytic therapies would be therapies that help break apart the thrombi or that will remove blood clots. So if you could imagine, if you are creating a different type of blood clot, then your normal strategies might not necessarily work. So here we show that ferric ions can activate, non-enzymatic blood coagulation. So without thrombin at all. So thrombin, remember, turns fibrinogen into fibrin, and the fibrin will precipitate and form this polymer. Well, if you just have iron ions, it can non-enzymatically cause that. So this is resulting in the formation of fibrin-like dense matted deposits, or DMDs, demonstrable by electron scanning microscopy. So this is just a type of form of microscopy or microscope looking at things. So these dense matted deposits are similar to those found in stroke and diabetes. So on the basis of these findings, we can conclude that the spontaneous formation of fibrin-like dense deposits in patients' blood may be a consequence of what is known as iron overload. So therefore, it is possible that the inactivation of unbound iron in the blood by small molecular weight chelating agents may prevent thrombotic consequences of the excessive accumulation of iron in circulation. So this is just another paper describing the fact that when you have free ferric ions in solution, and sometimes if you have the iron 2+, the iron 2+ can also oxidize into iron 3, so you can't really completely separate them. But if you have iron 3, you are going to get this basically precipitation of these blood proteins out of solution, and they are going to be resistant to fibrinolysis, or they're going to be resistant to the proteins degrading them, at least to a degree. So, knowing this, knowing all of this, if we step back, what can we think? So what I'm getting at right now is if iron is being increased in the system, it can be causing blood clots, and it can cause these blood clots through, say, interleukin 6. So let me go back to this paper over here, which is called Interleukin 6 and Hemostasis. So this is a review, Interleukin 6 and Hemostasis, and this is actually a very old article. It's from 2001. There's probably a lot more research that has gone into it right now, but I chose this because it has some nice diagrams that can help demonstrate how interleukin 6, which we know is upregulated by iron, how can interleukin 6 cause blood clotting? So here we can say we have injury or inflammation, and so things that could be injury or inflammation, this could be cutting, this could be the iron that is existing causing these oxidative species, this could be, well, the Galectin 3 binding protein and the Galectin 3 causing the white blood cells to dig themselves into the walls of the vein. All of those could cause this interleukin 6 production. So here they have the injury and inflammation is interacting or causing the endothelial cells to possibly produce interleukin 6. The injury and inflammation can also cause vasoconstriction, which is a different form of hemostasis. Hemostasis meaning the stopping of the blood, right? So you produce interleukin 6, which can then interact with the coagulation cascade, or causing these pro-coagulation factors from activating. And we'll describe those a little bit more in this other graph. And the interleukin 6 can also interact with the platelets, which can then form the platelet plug. And all of this can help with hemostasis, or things that stop the blood, such as clotting. So here, if we were talking about this coagulation cascade, figure 4, well, here is figure 4 that, this is 20 years old, so this is definitely probably needs to be updated. But I just want to show you just a quick overview of how interleukin 6 previously has been causing this. So interleukin 6 will interact with the tissue factor 7A, which can then it can do that, and then it can influence this cascade where it can turn prothrombin into thrombin. So that is one way that interleukin 6 can activate thrombin. Another way is interleukin 6 can interact with factor 8, and this interaction with factor 8 can also influence this cascade in another way as well. So those are two ways. A third one is through interaction with VWF, another protein. Interleukin 6 can also downregulate these anticoagulation proteins. So protein S can downregulate protein S, which then protein S normally downregulates factor 8, but if you downregulate protein S, then all of a sudden factor 8 is now more active, and so that can enter this pathway. Same thing with antithrombin. So antithrombin will prevent thrombin from working properly. Well, if you get rid of antithrombin, then you can activate this thrombin. And then the last thing that is described here is that interleukin 6 can create or cause the production of more fibrinogen, making more of the thing that generates the fibrin, right? Fibrinogen, making more of that can cause more clots. So here we can see interleukin 6, this pro-inflammatory cytokine, can cause all these things that can cause the production of more fibrin. Now, what does iron do? Well, iron will upregulate interleukin 6. It also will upregulate transferrin and ferritin. So know that that's also what's going to be upregulated in the presence of iron. But in the presence of these non-bound iron, it's going to upregulate interleukin 6 and cause all this to happen, and also cause the production of more fibrinogen. But what is unique about iron is that it doesn't need thrombin. Iron does not need thrombin to turn this fibrinogen into a clot. It doesn't need thrombin at all to cause a clot with fibrinogen. So basically, interleukin 6 is making all this fibrinogen as a result from iron, but it doesn't even need this thrombin. All of this is not even necessary to create a bad clot when you are just producing more fibrinogen, and then you have iron that's coming in here and causing fibrinogen to create its own clot with or without thrombin. So that is another thing that iron can be doing to influence this prothrombotic state. And so why or how do I phrase this? So what I'm sort of getting at right now is that I think that iron is going to, for one, upregulate, it's going to upregulate different proteins, and some of those proteins will be ferritin. So I will actually go over, let's look up ferritin. Sorry, I've been saying transferrin, but so you know, I don't know if I really should go too far into detail because this could get a little bit too complicated. Just know that transferrin and ferritin, well, transferrin can interact with ferritin, and those are both upregulated by the presence of iron in the system, or the higher concentrations of iron will cause more upregulation of transferrin and transferritin. We know that transferrin interacts strongly with Galectin 3. So Galectin 3 interacts with transferrin, and that could be one way that you can have the transferrin somehow influence the activity of Galectin 3. But not only that, Galectin 3 will also interact with the Galectin 3 binding protein, and that could also cause the white blood cells to dig themselves into the wall of the vein, which all of this can cause the production of more fibrinogen and cause the production of interleukin 6, which that whole system is probably going to put you into a procoagulant state. And then once you're in this procoagulant state, well, it's game over, because then all this iron is able to interact with the fibrinogen and then start clotting all over the body, which is what we see in COVID-19 patients. Now, how do I phrase this? Is this what's happening? I don't know. But there have been, at least, from the very beginning, there were some people were thinking that due to some of the proteins that are within the SARS coronavirus, that they are somehow able to interact with hemoglobin to release, or the beta chain of hemoglobin could be attacked by some of the proteins of the virus, and therefore cause the release of the iron from the hemoglobin. And one of the things that I think might be happening is that this is happening within the spleen, where the spleen has some ACE2 positive immune cells, and those ACE2 positive immune cells might be secreting these proteins into the spleen, where you have a very large concentration of red blood cells and old hemoglobin, and the cellular membrane of the red blood cells in the spleen are broken down because they're dying or they're dead red blood cells, which means as these viral proteins are being secreted by the white blood cells, they might be able to get into the hemoglobin and then release the iron from the spleen. And once the iron is released from the spleen, well then, holy crap, you have the labile iron. And this labile iron is, what's it going to do? Increase ferritin levels, it's going to increase transferrin levels, and the increase of those things might also be associated with the increase of interleukin 6, and then we also have an increase in the fibrinogen in the blood clots, and the production of these blood clots will also increase the D-dimer levels, which we also see is higher. Then the other things that we have to mention are the Galectin 3 binding protein and the Galectin 3. Those seem to be upregulated as well. Or I should say the Galectin 3 binding protein is upregulated as well, and that can also interact with all these other proteins that I've been mentioning. So this is why I personally believe that this is a necessary mode of research that we need to do. Go into this particular pathway with Galectin 3 binding protein, Galectin 3, transferrin, ferritin and interleukin 6, thrombin and fibrinogen. I think this entire pathway, or all of the pathways that are in between that, need to be targets for therapeutics. And so I was reading a paper earlier that was talking about how people who were on anticoagulants, even the full-fledged anticoagulants, were still experiencing significant blood clotting with COVID-19. And so this would not make sense unless the COVID-19 was using a non-traditional strategy to cause this blood clotting. And one of the non-traditional strategies would theoretically be utilizing iron, which can cause blood clotting without any thrombin whatsoever. So if you have no thrombin, it doesn't matter, clotting can happen. And even if you stop the interleukin 6 pathway and don't produce extra fibrinogen, well, even the fibrinogen that is just normally within your body could also coagulate out into blood clots, and it could also be resistant to the to fibrinolysis. So with that, I think I have made my point. I think I went over everything I wanted to. Oh, another thing that I might want to talk about later is that transferrin there is a lot of influence on transferrin and Galectin 3 production, and this is actually independent of iron. So if you insert transferrin that has no iron into creatures, that transferrin is actually able to cause the production of Galectin 3 with or without iron. However, the iron, if you aren't getting these injections of transferrin, the only way that you're going to make enough of this transferrin is if you have enough iron in your system, which is why, at least in this animal model, why it was necessary to have transferrin. And I think I will go over this paper tomorrow then, because I thought that this review was actually very interesting. So I think with all of that, I'm done. I think this was a very good way to, I guess, clear my thoughts as far as what type of pathology I'm seeing in people and how that can be related to cellular signaling, and how that can help us understand why there seems to be such a resistance from these anticoagulation therapies that COVID-19 patients are on. Some of them just seem to be completely useless. So this is why I think this was very beneficial and very edifying to just go through this thought process and get it down. So with all of that, thanks for listening and watching, and hopefully I will have another video out tomorrow. So if you have any questions, please leave them down in the description below, and I will have links to all of the papers that I have discussed in the description. So if you want to read those papers for yourself, especially if you have access to the papers, I highly recommend it. So with that, thanks, and I will see you tomorrow.